Okay, good afternoon, everyone. Thanks for the interest in the human genomics section. So I'm Aiping from Berry Genomics in China. So today I'm going to talk about long-read sequencing-based clinical applications for monogenic disorders with complicated molecular genetics. Okay, so three-tier program has been implemented in China for the prevention of severe and early onset genetic birth defects. So the rapid development of high-throughput sequencing has played a very important role for the prevention. So over the years, the prevention has expanded from chromosome disorders tested by next generation based NIPT, NIPT plus, and CMV-seq to monogenic disorders tested by like WES, other NGS panels, or even WGS. However, NGS cannot fully address those complex monogenic diseases with complex structural variations, repeat expansion, phasing problem, and the presence of pseudogenes. So there are about 400 monogenic disorders caused by genes with complicated structure or variations. So among those, like thalassemia, spinal muscular atrophy, congenital adrenal hyperplasia, and Fragile X syndrome are the top ones because the severity of the disease and relatively high prevalence. So in Berry, we have developed a long-read sequencing, a targeted long-read sequencing workflow, including reagent kit, instrument, and analysis software for these diseases. So basically, we get a genomic DNA from like basically all types of samples and use multiplex long-range PCR to get the target and do a standard PacBio library construction and then do the sequencing on SQL2 platform and then use in-house developed bioinformatic pipelines to code the variants. So our first clinical application is thalassemia, which is an inherited blood disorder characterized by decreased hemoglobin production. Symptoms depend on the type, and often there is mild to severe anemia. So as one of the most common autosomal recessive disorders in the world, thalassemia affects about 1 in every 100,000 individuals. So it's highly prevalent in Southeast Asia and Mediterranean areas. There are about 350 million thalassemia gene carriers worldwide. So thalassemia is highly prevalent in the southern cities in China with carrying rate of 1% to as high as 24% among different areas. Thalassemia is caused by imbalance between alpha and beta globins. So either impaired expression of the globin genes or overexpression of the globin genes will cause the imbalance. So it's very important to get precise genotypes for these genes. Alpha thalassemia is mainly caused by 3 large deletions and 3 SNVs in HBA1 and 2 genes, while beta thalassemia is mainly caused by 17 common SNVs indels in HBB in China. However, hundreds of other disease-causing variants have been reported, but not covered by conventional PCR-based or even NGS assays. So we have developed the targeted long-read sequencing assay called Comprehensive Analysis of Thalassemia Alleles, CATSA, which can detect deletions, SNV indels, structural variations in alpha, beta, and delta thalassemia. So which has a much wider detection spectrum compared to PCR-based methods and NGS. It can also distinguish between highly homologous genes like HBA1 versus HBA2, HBB versus HBD, and also do the phasing. So these are the two examples that showed the cis configuration and trans configuration between two SNVs. And the large deletions in the alpha gene locus, like Southeast Asia, 3.7, 4.2 kb deletion, Thai deletion, which is prevalent in Thailand, and field deletion, which is prevalent in Filipino. As well as these regional prevalent deletions of HBB gene. And also, this assay enables accurate detection of structural variations that caused by recombination between homologous genes, like alpha, triplication, Hong Kong alpha, and deletion and duplication in the HBB gene locus. So we have evaluated the assay for carrier screening of thalassemia by a prospective study of 1,759 samples from 10 centers. So for the common alleles, CATSA showed 100% accuracy, while PCR assay had 2 false negatives and 8 false positives. So CATSA additionally identified 34 positive cases and 3 couples with at-risk pregnancy. So we also identified 7 cases of alpha triplication that were missed by PCR assay, and 3 cases of Hong Kong alpha, which is commonly misidentified as 3.7 kb deletion by conventional assays. We also performed back-to-back comparison between long-read sequencing and the NGS assays. So the comparison was performed in over 1,000 retrospective samples. So from this study, NGS misdiagnosed 17 samples, including 13 with non-clinical significance, while the accuracy of the long-read sequencing assay was 100%. We also evaluate CATSA for prenatal diagnosis of thalassemia by studying by doing the testing in 278 amniotic fluid samples. So compared to PCR, CATSA has corrected the genotype and predicted phenotype in 8 fetuses, including 2 from beta thalassemia trait to intermediate due to beta-0 thalassemia compounded with triplicated alpha globin genes. Specifically, CATSA corrected 1 HPB variant from homozygous to heterozygous, which changed the predicted phenotype from beta thalassemia major to trait, potentially impacting the pregnancy outcome. So clinical chemistry, the journal where we published our work, has made editorial comment on clinical perspective on use of long-read sequencing in prenatal diagnosis of thalassemia, which I am quoting here. Though incremental, the increased sensitivity and specificity using long-read sequencing is an important advantage of this methodology in the prenatal diagnosis arena. So we also launched the Thalassemia Gene Atlas project, aiming to study the mutation spectrum of thalassemia in China. So this project tested over 5,000 samples with abnormal hematological parameters. And the assay was compared to a combination of assays, including PCR, MLPA, Sanger sequencing, and NGS. So within the detection range, the accuracy of CATSA was 100%, while the accuracy of PCR was 99.2%. However, a PCR has a very narrow detection range. So CATSA additionally identified rare variants in 156 samples, increasing the positive detection rate by 4.78%. Also, more than 20 novel variants have been identified by CATSA. So these are the clinical studies that for CATSA assay that has been published. And these are the novel deletions identified by CATSA assay. So this long-read sequencing thalassemia assay has really broadened the mutation spectrum of thalassemia. So in Barry, we have the reagent kit, SQL2 instrument, analysis software, automatic report system, and genetic counseling team. All these elements that are indispensable for large-scale clinical application. The first-time success rate of test is over 99.7% with turnaround time of about 8 days. So last year, we have performed the test for about 110 samples. And this year, we expect to increase the number to 1,400,000. So our second application is spinal muscular atrophy, which is a severe autism recessive neuromuscular disease due to progressive degeneration and loss of motor neurons. The incidence of SMA is 1 per 10,000 in live births, with a carrier frequency of 1 in 50. Due to the high incidence rate and disease severity, population-wide SMA carrier screening is recommended by ACMG to facilitate informed reproductive options. However, comprehensive genetic testing of the causal gene SM1 is really challenging because of the presence of highly homologous modifier gene SM2. So these two genes are 28 kilobase long, and there are only 5 nucleotide difference between these two genes. So conventionally, it's very difficult to discriminate these two genes. So in normal, a normal SMA allele mainly has 1 or 2 functional SM1 genes. And for SMA patients, both alleles have no functional SM1 genes. So for the carriers, 91% of carriers have 1 allele with SM1 deletion, and 4% has SM1 with disease-causing SMV or indels. And then there are 4% we call silent carriers. So those individuals have 2 functional SM1 genes, but on the same chromosome. So current SMA genetic testing methods such as qPCR, MLPA, and NGS focus only on the deletion carriers, so the 91%, while fail to identify carriers with SMV indels and silent carriers, accounting for 9% of all carriers. So we developed a long-read sequencing-based assay, which use long-range PCR to amplify the 28 kb SMA genes simultaneously to detect the copy number variation and the SMV indels simultaneously. So this is the first assay to achieve haplotypes of full-length SM1 and SM2, making it possible to detect silent carriers. So conventionally, SM1 silent carrier is deduced relying on an affected proband. So this long-read sequencing assay represents an effective and universal haplotype approach for SMA silent carrier screening through family tree analysis. For instance, in this family, so the father is a carrier with 1 SM1 copy, and the mother has 2 SM1 copies. However, this family has an affected proband. So from this, we can deduce that the mother has 2 SM1 on the same alleles. However, for the prevention of this disease, so our strategy is that when a partner of a couple is a carrier, the possibility of silent carrier of the spouse should be inspected using long-read sequencing with family trio samples. Like here, the proband has a big sister. So use father, mother, and sister trio analysis. We can see that the 2 copies of SM1 in the sister are inherited from the mother, which shows that both the mother and father are carriers. So in this case, prenatal diagnosis should be performed for the second child, or the family can seek for assisted reproduction technology for a second child. Alternatively, like father, mother, and the grandparents can form a trio for the analysis of silent carrier. So by this approach, we can get close to 100% of SMA carrier screening by long-read sequencing. Also, we have developed the assay for CAH, which is a group of autosomal recessive disorders due to gene defects in the enzymes required for cortisol biosynthesis. So the main type of CAH is 21HD, which is caused by the very complicated gene CYP21A2 because of the structural variation caused by the functional gene and the pseudogene. So this is the assay we have developed, and I'm sorry for the slides. So we have performed clinical studies for over 30,000 samples and addressed very difficult categories that have both deletion and duplication, which cannot be addressed by conventional MLPA and Sanger sequencing, as well as this category that has duplications and 2 heterozygous point mutation. So long-read sequencing enables the precise genotyping of these categories. We have also developed comprehensive assays for Fragile X syndrome, hemophilia A, and autosomal recessive polycystic kidney disease, which I'm not going to elaborate one by one today. So as a summary, we believe that incorporating these long-read sequencing assays into the current short-read sequencing dominated 3-tier prevention programs will facilitate more comprehensive and effective prevention of this disease and benefit the general population. Thank you.